Welcome back to Your Brain, a Personal Tour. This is Steve Soltwick. Today's seminar is about the decision to act. Whereas Hamlet had it easy, he only had to make a decision about whether to revenge his father or not. In everyday life, the decision to act is much more complicated. We're going to talk about the 4 ways your brain makes a decision. Some are pre-wired by Mother Nature. Some rely on mental time travel. And some are so habitual we don't even think about them. Each type gives us unique insights into our brain. So, hope you enjoy the seminar. Let's begin.
La noche irá sin prisa de nostalgia. Habrá de ser un tango nuestra herida. Un acordeón sangriento nuestras almas.
I want to frame the next 2 seminars with an analogy. Imagine you're walking in the woods and you come to a fork in the path. Of course, you want to take the path less traveled. So, how does your brain decide which path to take? That's the topic for today. Then the topic next week is, once you've made a decision, how does your brain remember which path you took? That is the topic of Seminar 3 next time. I've also added 2 books to the reading list. The first, Making Up the Mind, is a good general text for further understanding on how our brain constructs our mental world, including how different parts of our brain, say the visual part versus the movement part, may have different opinions on what the real world is. The second book, The Brain from the Inside Out, is a more technical book. I suggest you need some background in biology or other science disciplines to fully appreciate the text. This book, however, is written by the pioneer in the field and represents some brilliant and thought-provoking frameworks for understanding how the brain works in making a decision. So let's begin and get to the background of the opening quote. Amelia was the first female soloist to fly across the Atlantic. She was a best-selling book author. She was married to a famous book publisher, George Putnam. She was also a strong advocate for equal rights. Fierce determination marked Amelia once she had made a decision, and that's why she's on the slide today. She tragically died in 1937 on a circumnavigation flight around the planet due to a memory problem. But memories are in the next seminar. Let's talk about decisions right now. While Hamlet had it easy, life presents us with everyday demands that require decisions, and they begin at a very early age. Let's take some examples.
The marshmallow test is a really great way to show how children delay gratification. We tried it out with the 4 children we've been following since September 2010, Alfie, Millie, Mackay, and Pratmesh. Here's how it works. We had each child on their own sit at the table at a desk with a plate and 1 marshmallow. They could either choose to eat the marshmallow, the 1 marshmallow right then and there, or they could wait until I came back into the room and have 2 marshmallows. I left them alone in the room for 15 minutes. Take a look. The marshmallow test has been used for decades by psychologists. It's been used with children to predict later academic success, including literacy, SAT scores, and other academic outcomes. There's no definitive answers from the marshmallow test. It's not a matter of passing or failing. What we're looking for is whether children can really resist this piece of white candy sitting in front of them that's sweet, that, the smell of it, the allure of the marshmallow. In Pratmesh's case, we really saw this added curiosity because he had never actually tasted a marshmallow before. All of the children managed to show some level of self-control and resist the temptation to eat the whole marshmallow. As you can see from the footage, you can catch a glimpse into children's ability to control their impulses. This ability, which is developed around the time of kindergarten, can be linked to other outcomes later in life. At the end, the marshmallows were in kind of different states. Some had been squished, ripped apart, nibbled around, there was this temptation and there was this impulse to kind of try it out.
Now, this video is typically presented as an example of the delay of gratification, an exercise in self-control. I think now that you've seen what the children actually did, you'll recognize that they're deciding not to eat the marshmallow by deciding to do something else. Much of the socialization choices we are presented with in life are not simple delays of gratification. It's deciding to do one set of actions rather than another. Now, I said there were 4 ways that such decisions were made. Let's look at each one in turn. The first kind of decision is a reflex. It's a decision that Mother Nature has pre-wired into your brain because this decision is too important for your brain to deliberate. It's a stereotypical behavior, typically of a defensive nature, and it's designed to protect you, to keep you alive. But don't get an idea that only simple movements are reflexive. They can be quite complex. Now, one way a reflex comes into play is a reaction to physical harm. When one of your limbs hits a painful stimulus, the legs in this example, withdrawal reflex, rapidly moves the limb to minimize harm. In this diagram, we've just stepped on something sharp. There's specialized receptors called nociceptors that detect the harm, transmit a signal to your spinal cord, and via a pre-wired path adjust your body to withdrawal. First, muscles are both activated and relaxed so that you can bring your foot away from the sharp and painful stimulus. But notice what also happens. Your other leg is simultaneously prepared to handle the shift in weight. Your brain is informed, but it will take a bit of time. You're going to feel the pain long after you've moved your limbs. So you don't think, you react. And most of the time, that's a good thing. Now, don't get the idea reflexes are just for jerking your limbs. They can be whole body, like sometimes we'll see in this clip. And they can be very, very practical. In this clip, we see how an enterprising vet uses a binder clip to freeze and calm a cat. The cat's on the examination table. The effect is dramatic and harmless. The cat is not traumatized in any way. When we take the clip off, he does what a cat does, which is look around and then jump off the table. The reason this hack works is that it activates a reflex in the cat from his kitten days, when he was carried by his mom by the scruff of his neck. Now, my wife and I are dog people. Do not try this on a dog. But for the cat people in the audience, we'd love to hear your experience with doing this. Reflexes can be even moral judgments. Here we study the actions of a 6-month-old infant to the action of geometric shapes. Now, the shapes must have eyes for this experiment. That's important. The infants watched a plucky circle try to scale a mountain. A helpful triangle, not shown, was helping the circle to get to the top. But a mean old blue square pushed the circle back down the hill. Infants reliably reached for helper geometric shapes rather than hinderer geometric shapes, thus performing what many would say is a moral judgment of character. But wait, it's a little more complicated than that. In this experiment, 9 to 14-month-old infants were observed to record their preference to either graham crackers or green beans. I know what you're thinking, but 47% of the 9-month-old preferred green beans. By 14 months, only 31% liked the green beans, so they're getting smarter. Then, infants observed 2 hand puppets expressing food preferences, crackers versus beans. When the infants were then given a choice of reaching for one of the puppets, they invariably chose the one with the same food preference. In other words, they preferred the us of the puppet liking a similar food. Then, helpful or hurtful shapes with eyes were brought into the experiment, and the shapes interacted with the puppets. Babies love seeing a food us being helped, and conversely, a food them being hindered. So this is not about infants preferring helpers. It's about preferring agents that are similar to them. and preferring agents that hinder puppets that are different from them. We'll talk about the implications of this later. Now, how to make sense of this somewhat surprising results on, quote, simple reflexes. The theory I subscribe to is that Mother Nature endows us with a pre-wired emotional set of reflexes, kind of like the automatic mode on a camera. A simple, quick action that's appropriate to probably 80% of our situations. As we age and encounter more complex and more social situations, our brains have evolved a more advanced system, kind of like the manual mode of a camera. The advanced systems are really good, but they must be consciously and deliberately applied. We'll study those systems in just a bit. Let's let Homer take a crack at summarizing this session. The first thing Homer says is that reflexes are a do-it-now. They're time urgent, no time for deliberation. They're very useful in avoiding or reacting to harm or threats with an automatic range of behavior. Those behaviors can be simple as a knee-jerk or as complex as a moral judgment. However, reflexes can also be overridden, kind of like an advanced manual mode. We'll see more of that soon. This is especially important when reflexes seem out of place in the modern world. Now let's take a look at our second type of decision. We'll call such decisions associative decisions. These decisions unleash a reflex-like behavior, but from a learned association. An association is a mental connection formed from learning. The science of learned associations is uniquely associated with Ivan Petrovich Pavlov, shown with a beard here. Dogs were measured in their saliva response to food, which reliably elicited salivation, or to a light or tone, which did not elicit salivation. When the light or tone predicted the delivery of food, the dog salivated to the light. A learned association drove the decision. Such learning is often called classical conditioning or Pavlovian conditioning, and the decisions are termed associative decisions. They represent a fundamental type of decision making by your brain and are actively studied even today. And I must also note that my wife, Becky, is a professional dog trainer, so I'll know she'll be very proud of me getting dog video into these slides. Now, the dog of research today is the California sea hare. You see two of them here. These animals are they have very simple behaviors. They basically move around in the sand. They squish water with a siphon. You'll see that disturb the surface soon. And they have a gill to breathe. These animals are the perfect research animals for this type of decision learning. They have illuminated the exact mechanism of how all brains learn such behavior. And they've earned a Nobel Prize in the process. Well, not these two hares, but the scientist, Eric Kandel, who conducted the research that got the prize. The scientific name for this dude is Aplysia californica. And since he's from California, we're going to give him some cool sunglasses. But really, he's cool for one unique aspect of his brain. His brain has only got about 20,000 cells compared to the 86 billion in your brain, and those cells are huge. Many are visible to the naked eye. Neuron R2 is over 1 millimeter in diameter. It's roughly the size of the B on the Liberty Penny. These individual neurons can be uniquely identified visually. That means we can experiment with multiple different California sea hares, but study the same neuron in each individual as they learn and express an associative decision. Let's teach this dude a new trick and see what happens in his brain. We're going to use two stimuli. One will reflexively cause a behavior and is called an unconditioned stimulus, or US for short. In this experiment, it's a mild shock to the animal. Our guy will reliably retract his siphon to this, the US stimulus. The other stimulus is touching the animal. Normally nothing would happen. The guy would leave his siphon out and about. But if we pair that touch, called the conditioned stimulus, or CS, with a US of shock, we'll find that after training, the animal has a vigorous withdrawal of the siphon to the touch. The animal has decided to withdraw his siphon since it's associated with the shock. The key results are shown in the bottom right of the slide. Since we can see the brain cells of this guy, we can watch what happens to the exact neuron that underpin this change in behavior. We'll show the schematic of these brain cells in the top right. The L29 neuron on its own can activate the motor neuron which withdraws the siphon. That's how the shock causes the siphon to withdraw. A sensory neuron corresponding to the touch CS also converges on the motor neuron, but first that connection is not strong enough to evoke a withdrawal response. Now we know that after training, the sensory neuron can activate the motor neuron to withdraw the siphon, so what happened? Well, this happened. We've expanded our view of the connection between the sensory neuron and the motor neuron. What we see is the creation of a new path in the brain. This path is a combination of two changes. There's a short-term change where previously empty legs of the sensory neuron became filled with additional packets of neurotransmitter, the chemical that a neuron dumps on another neuron to get it to become active. Now all of a sudden the motor neuron is affected by the sensory neuron and the siphon begins to withdraw. Over the course of several hours, about 12 to 18 hours, a dramatic event happens. There's actual growth in the brain. Now, new legs appear on the sensory neuron and they become populated with the neurotransmitter. There's a long-term structural change in the brain that reflects this learning. Should we say a memory? This change will now evoke the decision to withdraw the siphon to the tactile CS. So, short-term, there's a transient chemical change, followed by a long-term permanent structural change. Wow. So if our brain grows as we learn new information, does that mean we need a really big skull to be smart? Well, Star Trek writers sure think so. And in normal life, the skull actually does grow dramatically to accommodate a rapidly learning brain, but only to a point. Over the first 3 years of life, the skull almost triples in size. Man, that's a lot of learning, as any parent or grandparent can tell you. But after that, the skull size is relatively constant. So then how do we learn new things if there's no room in the skull for increased brain circuits? Well, that's a problem for memory, which we're going to investigate the next seminar. These same changes appear in other organisms, like rats and like us. Those kinds of changes are important to understand. Let's take an example. We already know that a rat will freeze if you threaten her, say with coyote urine. She'll also freeze if you shock her foot. The pathway in the brain for that foot shock is shown on the right under the US label. Sensations of the shock go through a couple of areas of your brain and then concentrate onto an area of the brain called the amygdala. The amygdala is a specialized structure in the mammalian brain that detects, among other things, items that represent a threat to the organism. When the rat gets a foot shock, the sensation travels through her brain to the amygdala, which drives a behavior of freezing. But freezing isn't the only thing a rat does. There's internal changes to the rat. Her blood pressure goes up, stress hormones are released into her blood system, etc. After we associate the tone and the shock, the tone will elicit the external behavior of freezing, but also changes in the internal state. Blood pressure rising, release of stress hormones, etc., etc., etc. In essence, the rat has an emotional response to the tone. Why do I say that? Research has shown that emotions are responses to internal bodily changes like heart rate, stress hormones, and other things. You see this in the woods and bam, your amygdala is going crazy. It will prepare a flight or fight syndrome. Your blood pressure is going to go up. Stress hormones are going to be released into your bloodstream. Your heart is going to race. Based on a long set of psychological research, it is because your blood pressure is going up, because your stress hormones are being released, and because your heart is racing that you will report feeling fear. Stated another way, we feel fear not because we're looking at the bear, but because we're running from the bear. So this whole set of associative decisions is part and parcel of how we learn emotional responses to various stimuli. These emotional responses are automatic and they come as part and parcel of the associative conditioning process. And that's one way that emotions get attached to various things in our life. This concept of emotion can be used for treatments other than scaring people in the woods. It turns out that about 20% of the troops returning from Afghanistan, Iraq, or foreign service suffer from PTSD. Standard therapies only result in about a 60% remission. The many members of the Special Forces report hypervigilance and anxiety, similar to the effects shown in the movie American Sniper. Well, if you block a specific autonomic nerve center, called a ganglion, with a pain blocker, there's widespread reports of immediate and profound PTSD relief. One shot seems to block the emotion-generating aspects of high heart rate, fast breathing, and muscle tension. In other words, blocking the physical attributes that underlie emotion seem to release the perception of stress and allow therapy to progress. More recently, research seems to indicate that the stellate ganglion blocks used in PTSD here can even be effective in relief of certain impacts of long COVID, for example, the loss of smell. Now let's ask Homer what he thinks about this whole set of associative decisions. Homer has 2 points to make. One is that even slimy sea thingies can learn to predict the future. Take an innocuous stimulus like a touch and pair it with a shock, and even a California sea hare can see the future. It will do something in anticipation of that shock, and that's an associative decision. The second thing Homer wants to say is that those kind of associative decisions change your brain, and it changes your brain with new growth. That structural change, as you learn things, has several consequences. As we know, your brain is fully encased in bone. That means it's a fixed volume. If every time you learn something and there's new growth in your brain, sooner or later you're going to run out of room. How does the brain deal with that? Well, it turns out that one of the prime functions of sleep, and we're going to learn about that more later. Right now, we're still going to explore, complex decisions and how does your brain make them. Well, those kind of decisions are called deliberative decisions. They're the kind of decisions where we have to trade off priorities or options. We could do one thing or another, and we evaluate the options before we actually decide and commit a particular behavior to action. Believe it or not, even our little friend the rat here goes through the same events in his brain as we go through deliberative decisions in our brain. We're now going to see the rat's brain go through, such a decision, and what is he thinking in terms of mental time travel as he makes that decision. And what goes on in a rat brain goes on in your brain. Both our brain and the rat brain have structures that help us perform these kinds of activities. And to really get a good handle of them, we're going to have to look at actually 3 different places in your brain. So let's take them one by one. The first brain area I want to talk about is called the hippocampus. We see it here in a rotating model of your brain. It's the highlighted linear structure at the base of each hemisphere. It's about the size of your little finger, and it's amazing that such a small part of your brain is so important in the way we make decisions. The hippocampus came to prominence when this young man, age of 27, received an operation in 1953 to cure his epilepsy. The operation removed his hippocampi and other parts of his temporal lobe. The operation was very effective at curing his epilepsy, but it came at great cost. In essence, this person lost the ability to form new memories of conscious experience. While he was alive, he was known as H.M. He died in early 2000, and we now know his real name was Henry Mollison. Henry was well aware of his memories of childhood, but every time he met his doctor, he had to be reintroduced. He remembered the doctor's name during the course of the session, but the next day, he had to be reintroduced all over again. Now, Henry could learn certain memories. For example, he could learn drawing and coloring tasks, so he has what is called procedural or motor memory. But making memories about his experience, something called episodic memory, and using those memories to make decisions was way beyond Henry's capability. Events like these brought the hippocampus into the crosshairs of intensive research. Now, the hippocampus had been known to neuroanatomists since the 16th century. In fact, the first guy that described the hippocampus actually thought it looked like a silkworm. The hippocampus from a human brain is shown on the left here, and the nice little cuddly seahorse is shown on the right. The neuroanatomist changed his mind and said after further inspection, it does look like a seahorse. So, being a good neuroanatomist, he forged a name from the Greek, and voila, we got a new name for a very, very important part of your brain. Fairly recently, there's been a major discovery about the hippocampus, and that is the discovery of hippocampal place cells. These are cells which are like GPS cells. After a rat has explored some space like this S-shaped track here, you'll find brain cells that are active when the rat is at a particular point in space. For example, if we record the neurons in this rat's brain and we let him run down the track, when he gets to this yellow section of track, we could see specific neurons become active. The neuron's burst of activity might sound something like this. And then later on, when the rat got to the orange part of the track, a different set of cells would burst again. So there'd be a population of cells within the rat's hippocampus that would preferentially burst when the rat was at a particular point on the track. By convention, we usually color the neurons' activity. They're not really orange and yellow, but it helps our understanding of what they do if we color them uniquely. I assert when the yellow cells are firing, the rat is thinking about being in the yellow portion of this maze. When the orange cells are firing, the rat is thinking about being in the orange portion of this maze. Now, isn't that amazing? Let's see it in real time. What we will see here is an actual recording of the hippocampal place cells as a rat explores an M-shaped elevated walkway. The position of the rat is going to be denoted by the white light. He's connected to a wire that allows us to record and amplify the neuronal signals of about 10 individual cells. The firing of those cells will be denoted on the left-hand side of the maze. We'll put a little vertical tick mark whenever the particular cell fires. The cells are color-coded so that we see the map, so that we can map the activity of the cells against where the rat is in space. Let's see what happens. I'll let the film loop a couple of times and I draw your attention to the following. Note how the rat is going first up and then left and then down. That's his path through space. As he negotiates this space, the cells follow a particular pattern. The blue cells fire first, then the kind of greenish ones, then the yellow ones, then a few reds, and then finally the pinks when he finishes. This sequence of colors maps to the rat's path through the space. Knowing which cell is firing, we can place the rat in space. Knowing the sequence of cell firings, we can map his path through space. We can use this information to see a rat think. It turns out that hippocampal place cells fire at times other than just running through a maze. And that fact is going to help us understand how a rat thinks about a path. That other time hippocampal place cells fire in order is during something called a sharp wave ripple. This ripple is a very short event, only 1/10 of a second long, but during those sharp wave ripples, we see place cells firing in sequence: purple, blue, green, yellow, pink, then brown. We know that place cells actually map to space, and if we plotted those to space, we would actually trace out a path. The sharp wave ripple shown on this slide is the rat thinking about moving through space. Down, right turn, then up. The ripple is only 1/10 of a second long, much too fast for movement to occur. This is the rat thinking. This kind of thinking is an essential underpinning of deliberative decisions. The second area of the brain we need to talk about is a time-traveling reward system. The reward system of your brain is actually tied to a specific neurotransmitter. Neurotransmitters are those chemicals that one neuron dumps on another to influence the behavior of that neuron. There's several different kinds of chemicals, but the reward system of the mammalian brain uses a specific neurotransmitter called dopamine. Often the reward system is called the dopamine system. What do I mean by a reward system traveling in time? We'll look at 3 different events. The first event is represented by the neuronal histogram at the top. This shows a record of brain neurons in the dopamine system and how they're firing as things happen. In this experiment, a light is turned on randomly and food is placed into the cage randomly. There's no association between the light and the food. You'll notice that the dopamine system doesn't do anything to the light, but boy oh boy does it go crazy when a reward is dumped into the cage. Remember, your brain has no direct sensors, so it needs an internal representation of reward, and the dopamine system seems to do that. In the middle tracing, we've paired a trigger to that reward. The trigger predicts the reward. Notice that the dopamine system migrates in time and now reacts to the earliest event in time that predicts the reward, in this case, the trigger. It goes off on the trigger, and note that it doesn't go off on the reward. The reward is totally predicted by the trigger, so the neuronal activity moves in time now to go off on the trigger only. The bottom tracing takes it even a step further. We now have an instructional signal, something that basically comes on to tell the animal, oh, by the way, the trigger is going to come on in 1 second. The trigger comes on and is then followed by the reward. Look at what the reward system does. It goes off again to the earliest predicting signal, in this instance, an instructional cue. You'll note that the vigor of the response decreases over time. The most activity for the reward system is the actual reward. Something that predicts it, or something that predicts something that predicts it, will also cause the system to activate, but in an increasingly smaller and smaller levels. Right? This shows the reward system of your brain can not only represent a reward, it can travel in time. Like the place cells representing mental time travels of space, the dopamine reward system can represent mental time travel of reward. The third and final part of the brain we need to talk about before we put it all together is something called the basal ganglia, shown highlighted in this rotating view of your head. The basal ganglia sit in the middle of your brain. There are multiple individual areas, but we don't need to go into that right now. We'll just consider them as a whole. I want you to think about the basal ganglia as the go-no-go system of the brain. Various potential movements or behaviors compete in a race condition within the basal ganglia. The first one that gets to a threshold is committed to behavior. The other potential behaviors are suppressed. Let's see how that works. We're going to consider two potential actions, turning right or turning left. We're going to look at a slightly different metric of neuronal behavior called a heat map. In a heat map, the color relates to how many neurons are active and excited. The warmer the color, the greater the activity. Deep reds mean lots of neurons are firing. Deep blues mean very few neurons are firing. In this diagram, we map the neurons in the basal ganglia associated with either turning right or turning left. Each trace is aligned with an arrow that points with time. There's probably 100 or so neurons represented here, and we're following them all in time. We see many significant phases. The first is both potential actions show activity early on. Activity builds for both choices until one of the actions crosses the threshold. Once it crosses that threshold, that potential action is committed to behavior. It will take some time before the behavior is actually expressed. Movement objects must be formed, signals have to be sent out to the muscles, etc. The prime point I want to make with this slide is that 1, potential actions are all simultaneously active. 2, one action gets to a threshold before any other. And 3, that threshold crossing action is then the one committed to behavior and expressed. Now, I also draw your attention to the non-expressed movement. It goes very, very blue. That means that the potential action is being suppressed. That is a hallmark of this system. One potential action hits a threshold, it's committed to eventual behavior. The other action is suppressed. This effect could be important in obsessive compulsive behaviors, and I'll have a bit more on that in a minute. But right now, let's just keep it simple. The main points are multiple actions being represented, one hits a threshold, and that's the one committed to behavior. Let's put all of this together and watch one of our favorite rats as she moves to a choice point and makes a deliberative decision. In this maze, choosing the green path has always consistently been paired with food. Turning to the right is the path to reward. No food has been paired with turning to the left. That's the reddish path. What's going through her brain as she gets close to the choice point of the maze? Well, her hippocampus, using sharp wave ripples of place cells, mentally travels down each choice path. Turn right, and there's food. The reward system, which can also travel in time, evaluates that thought and produces a burst of activity, which is then fed to the basal ganglia. Her hippocampus will also mentally time travel down the left path. No reward, no burst of activity, nothing is fed to the basal ganglia. The basal ganglia as a group evaluates turning right versus turning left until one action exceeds a threshold. Turning right ultimately gets to the threshold, the activity of the reward system and the basal ganglia, and then gets committed to behavior. A movement object will be expressed, muscles will point her towards the right, and she'll scamper down the right-hand part of the T maze, and the left-hand turn will be suppressed. Isn't that awesome? Let's see some of these cells in actual operation. We're viewing a rat in a self-returning maze from above. He'll run down the center part of the maze and then make a right or left turn, and then come back up to the top. He'll run down the path again. The film has been edited to show alternating choices, first to the right and then to the left. We'll look at some cells that are active as the rat is deciding what to do. See if you can use the actual activity of the cells to predict which way the rat is going to turn. Listening to those cells, I hope you reached the conclusion that they were active and bursting when the rat was going to go to the left. Behavior of these cells demonstrate what we talked about today, the rat making a decision. Remember in the introduction when I said you think like a rat? Well, here's an example of a research report that shows that. The sharp wave ripples that we see in the rat are also present in humans, and really a lot of other species as well. In this particular experiment, the ripple was not only associated with making a decision, but in the memory of an object. We're going to see more of our friend the sharp wave ripple in the next seminar on memory. It's time for Homer to join us again and talk about the two main points of this section on deliberative decisions. Homer, in his own unique way, says even rats have a brain that can travel in time. There turns out to be a fundamental importance in the way we understand our brain. Our brain can move us around in time and space, just like a rat's brain can move them around in time and space, both for physical space, for time and reward. Activity from those events can then feed other systems which can determine which particular potential action is going to be expressed in the movement of muscles. But also, Homer has a second point. And if you know anything about Homer, you know he's a bit of a lazy guy. So what he says sure seems to take a lot of effort in making this kind of decision. And he's very, very right. Your brain takes 25% of the caloric load of your body. In other words, 25% of the energy in your body is spent on the working of your brain. Mother Nature is very careful about how you spend energy, so that brings us to the fourth kind of decision. Habits. Habits are decisions your brain has delegated and is really not that much engaged in anymore. Habits occupy an amazing amount of time, and it turns out they're quite useful. Almost half of the behaviors you do on any given day are habits. I bet you didn't know it, but there are psychologists who measure how we spend our day, and research shows that about 45% of the behaviors that we do on any given day is a habit. In nature, the percentage for wild animals can be even more. If you don't think you're a creature of habit, try changing your shower routine. Your shower or bath routine is a highly stereotypical behavior of the sequences, for many of us. That means we really don't deliberate over each individual behavior we're doing. Each behavior seems to be cued by the behavior just before. First wash this bit, then that bit, then the other bit. We tend to have such rigid routines when we bathe, when we drive to work, tie our shoes, and other things. We are all creatures of habit. The benefit of habit is reduced energy load on the brain. This reduced energy load is particularly important to the dopamine system of the brain. The dopamine system is an extensive system of neurons in your brain that is sensitive to surprise. Your brain is very active when this system is engaged. The origin of the system is shown in the lower right. It's in your brainstem, the ancient part of your brain. This part of the brain controls breathing and autonomic functions, things that you don't have to pay attention to. Output from the system innervates wide-ranging parts of the front part of your brain, the frontal cortex. This is where outcomes are evaluated. So when this system is surprised, it activates huge portions of your neocortex, and your brain is chewing up energy like crazy. Now, why the system is active has come under intense discussion these days. Traditionally, the system was thought to be the reward system. When the system was active, the brain was signaling a reward to other parts of the brain. However, the system acts to predict the reward as well, as we just saw. And finally, there's a new set of results that indicate the dopamine system may actually be the causal association system. Let's look at that in a little more detail. We're going to take a simple Pavlovian learning situation where a bell indicates not dog food, but good food, like ice cream. The question is, how does the brain learn that the bell predicts the ice cream? There are a traditional model where the dopamine system is associated or assumed to predict the outcome and then learns when the results are surprising. But that's not the only way the system can learn. The dopamine system could be signaling, hey, dummy, you should remember what just happened before this great ice cream got delivered to you. In essence, the system is signaling that a meaningful outcome just happened. Your brain needs to remember it. Now, the two models make different predictions if you manipulate the system, and that's exactly what the researchers did 11 times. The results supported the causal detection model consistently. Now, this is a surprising and exciting result, and it needs more study, and I look forward to future research on this topic. Regardless of which model is true, it would be a clear evolutionary benefit to manage the energy load of your brain. One strategy your brain could use is to use the simplest set of stimuli in the environment to make a decision. Like in your shower routine, you just use a simple movement, washing your hair, to cue the next movement, washing your face or whatever. Is that really what happens in real life? Well, yes, and we can show that experimentally. The experiment deals with a rat in a maze. Surprise, surprise. It helps to imagine the maze labeled with compass points. But before we describe the experiment, I want to ensure you have a practical view of the maze itself. It doesn't look like this thing on the left. It looks like this in a room. Now note, the rat can see all sorts of stuff in the room, and the rat could use them as signposts in making a run. So from the rat's perspective, the rat may not be turning to some sterile point of the compass to go west or east. The rat could be turning to the red chair or some other landmark in the room. This perspective is important in understanding what happens next in the experiment. Now we're going to train the rat that food is always in the east. So if we start the rat in the south part of the maze, she would run down, turn right, and enter the east section of the maze and get the food reward. Periodically, we're going to probe how the rat is learning, the start location. We'll start her in the north side and then observe which way she turns in the maze. The rat can use two strategies in deciding which way to go. One strategy, known as the place strategy, means that the rat will use the cues outside the maze, like chairs in the room, that kind of stuff, to deliberate and determine which way is east. She will then go that way. If she started in the north, she will turn left to get to the east. If the rat is using what is called a response strategy, she's going to do whatever the response she's done most of the time in training. In other words, turning right. In this instance, starting from the north, she would turn right and wind up in the west side of the maze. What does she do in training? Well, it depends on whether the habit has been formed. Early in training, when the rat is just initially learning, she takes a place-oriented decision. Now that's a deliberate decision and runs to the east. Why? Because early in training, her reward system has got her brain lit up, Her hippocampus is moving around in time. The basal ganglia is integrating information, with the frontal cortex. The frontal cortex is evaluating outcomes. This rat is fully aware and processing all the stimuli in the room. Her brain is chewing up energy, and she goes to a place, the east. Then if we overtrain, running down the maze becomes almost automatic. Now, when we start her in the north, we find the rat takes an associative decision. She just turns right, which has been paired with finding food so many times, the rat is not thinking. She's running down, she gets to an intersection, and the normal thing to do is turn right. And that's what she does because it's a habit. The rat is really only using a small part of her brain. Part of the basal ganglia called the caudate nucleus, and that's shown in green here. The point is she's not really processing many stimuli. She's running down the maze. Running down the maze means turn right to get food. So from an energy use perspective, and from showing us how habits form, this experiment illustrates exactly what happens in the formation of habits. We go from where our brain has been awoken and lit up by surprising events, and where we're fully engaged and cognizant of all the stimuli around us, to only using a small part of our brain to figure out what to do next when we get overtrained on the task. As beneficial as habits might be for the energy utilization in your brain, they're also the source of much human and hamster misery. Because habits, when formed, are very, very difficult to break. Your brain's on autopilot. Habits can be maladaptive, and it's very, very difficult to break chains of autopilot behavior. Because your brain's really not evaluating other options. It's not deliberating anymore. It's performing automatic associative decisions where one step leads to the next, and at the end of some of those chains, there's not a good place. There's another aspect of habits to cover before we close. Recall how the go-no-go system builds activity in a decision up to a point where the action is selected. That's shown in red here. But look at the blue. Other behaviors are suppressed. In other words, doing an action inhibits the likelihood of doing some other action. Where that might be related to habit? Well, obsessive-compulsive behaviors. This go-no-go system has been implicated in this syndrome, and a better understanding of it in operation will point the way for therapies to reduce the suffering here. Now, Homer's going to weigh in at the end of this section and give us two observations of note. The first is Homer really likes habits. They help his simple brain to not get too tired. Homer is not one to really apply himself most of the time. Homer is absolutely a creature of habit. He manages his brain at a very low energy level, and that's just fine for Homer, and wish him well with that. But Homer also notes that once bad behaviors turn into habits, they are rigid. They become very difficult to change. That's one of the challenges of modern behavior, and I think Homer's got it nailed here. The study of habits and the way of breaking those habits can offer great hopes to segments of our population who are truly in need. I'd like to close our seminar today with some overriding thoughts. First, 3 of the decision systems we've discussed today, reflex, associative, and habit, are all automatic. Your brain is wired to do this. Yes, you can override reflexes, and there are ways of modifying habit, but in general, these decisions and systems are automatic. They reflect the way your brain has been wired by Mother Nature. They're not going to change for thousands of years. Most of the time, say 80% or so, they seem to work well. But when the ultimate behaviors out of these systems are ill-suited for the facts of modern life, it's not necessarily because people are evil, it's because their brains are wired that way. To make progress, we need to switch to manual mode. This is a deliberate choice or a decision to override what our automatic brain systems are doing. We can use these deliberation systems to address automatic bias and other kinds of associations that our brains naturally make. One example comes from modern symphonies. People generally tend to pick groups that are similar to themselves, in other words, us groups or in groups. So if white males already in the symphony are choosing, the auditioning players, white males are going to tend to pick white males. Modern symphonies have found that blind auditions accommodate and address such automatic bias. Performers audition behind a curtain before the evaluator, therefore the evaluator doesn't know the gender, doesn't know the race, doesn't know the ethnicity of the performer. This has turned out to be a wonderful way of eliminating gender and other kinds of bias. A similar process is now being used by NASA to allocate time on the Hubble telescope and others to eliminate gender, ethnic, and university bias. And then finally, this process is being tested in the legal system to address hidden bias. The key thought is to use our deliberative brains to override our bias brain. An afternoon of training is not going to eliminate the evolutionary skill of hundreds of thousands of years. The only other parts of our brain, devising new modes to counteract a natural bias, is going to address the situation. I'm glad we're starting to make real progress using such approaches. This concludes Seminar 2, The Decision to Act. I hope you enjoyed it. Let me foreshadow the topic of Seminar 3, The Structure of Memory. Memories are complex, multidimensional, slippery concepts that have several processes within the brain. They also have a structure within the brain. We'll talk about that in the next seminar, about the essential characteristics of memory. One type of memory is like a story from Proust, the French writer. Proust wrote 7 volumes and thousands of pages about a memory that came from a fatal encounter with a famous Madeleine, a cup of tea on a cold winter's day. His memories came out in rich, detailed patterns. However, this is but one way memories are stored. Are there others? Well, yes. And there are different brain structures which bring them about. A good analogy is the totem poles of Pacific Northwest Indians. Totem poles are not an elaborate reconstruction of a story as told in the pages of a book. They're the essential elements of the story as carved on a totem pole. To give a familiar Western example, the totem pole of Goldilocks and the 3 bears would be a pole that had a bear, a bowl of porridge, and a bed carved on it. The story would be reconstructed from these symbols. Now, of course, that means that 2 different people might tell the story 2 different ways, or the same person on 2 different occasions might tell the story again in 2 different ways. We'll see how memory structures relate themselves to brain structures and underpinning a Proust-like story and a Pacific Northwest totem pole. Please join me next time to see the fascinating story of the structure of memory. This is Steve Saltwick. I'll see you in the next seminar.
Ciégame, mata mi corazón.